Okay,
All right,
maybe okay
is this
now.
better? Can I be heard now?
Yes.
All right. One might think after all of this experience with Zoom, we'd be good at this by now. Ah. Yeah. Speaking of dual tasks. Yeah, so I have I have given a version of this talk before. If you've seen it, then I hope you will have excellent questions, either while I'm talking or at the end. I'm giving a talk which is relevant to the clinical focus of this session in terms of objective measures of speech intelligibility, but we'll just have to get there a little bit. I'm trying to expand the barriers here, or expand where we think about measures of speech tracking. This group, the CogHear group, was initially very focused on auditory attention detection, and that's still of great interest. I think this week we're going to be hearing about a lot more topics of that, but a lot of what I'm going to talk about is related to different aspects of speech tracking, which I think are really can be used and broadening our view of what speech tracking means. The title, Neural Representations of Same Species Vocalizations in a Human Primate Model. Well, of course, hey, I'm hitting the right button. There. Let's try that again. In humans, because this is a human primate model, but I will be talking about non-human primates in general. So I wanted to sort of bring up this tension between how do primates hear, how do humans hear, which are a kind of primate. And then if we're going to talk about same species vocalizations in humans, then that's really just speech. So I am going to be talking about how human brains process speech, but in the context of how other animals process speech and how they process same species vocalizations. So I do like this original title better. In case I get too fast at the end, these are everyone I want to give thanks to, both individuals and organizations for funding and support. The first part of the talk will be an introduction, that the human auditory system is a mammalian auditory system. Most mammalian auditory neurophysiology recordings are location specific. You plunk the electrode in the area of the brain you want to record from. Most human auditory neurophysiology recordings are whole brain. EEG and MEG are the ones that I'll be talking about today. I will not be talking about ECOG and related methods today, but other people this week will be talking about those. After that, after the introduction, then I'll get to neural processing of same species vocalizations versus the neural processing of speech, because it depends which species we're talking about when they're processing speech or processing same species vocalizations. And thinking about speech as a vocalization that is also a carrier for language. Many speech tracking experiments really don't use the language. They use just the vocalization part. And I want to broaden that to the language aspects. And also I want to talk about categorical perception and also neural processing of categorical perception in vocalizations in speech. Categorical perception is an important part of language, but it turns out that it's also an important part of same species vocalizations not for humans, and even an important part of neural processing of speech not for humans. So I just want to broaden all of these categories. So let's talk about mammalian auditory brainstem at first. This is the figure on the left is similar to one that Sandy showed earlier. One can record from the cochlea, the auditory nerve, the cochlear nerve, different parts of the cochlear nucleus, the inferior colliculus, midbrain. When one is inserting electrodes into these regions in a non-human prep. When we're talking about looking at auditory brainstem responses in humans, we will instead look at things much more like the graph on the right, which doesn't show where we're recording from, but instead shows a single recording, but with specific peaks at specific latencies. This is called the auditory brainstem response. It's typically recorded in response to a punctate stimulus, such as a click, or actually many, many repetitions of that click. It's characterized by 3 robust peaks, frustratingly named peaks 1, 3, and 5. There are peaks 2 and 4, but they are not robust. Wave 1 comes from the auditory nerve or the cochlear nerve. Wave 3 from the cochlear nucleus, from the ventral cochlear nucleus. And wave 5, the largest of them, comes from the inferior colliculus, from the midbrain. So when we talk about where a response comes from in the auditory brainstem in humans, we're really talking about when we see the response. At what latency do we see the response? We're using latency as a proxy for location. And that stands up well. It can't solve every problem, but it can solve a whole host of problems. Now, this turns out to be also true when we use continuous speech. So on the right, we have a figure from Sean et al. I can't get rid of this little thing. It's Sean on the lower left. This is on the right-hand side are temporal response functions, TRF. I'll talk a little bit more about TRFs in the next few slides, but it's obtained by deconvolving a response, the response with the stimulus. These stimuli are continuous speech or music. These stimuli are 60 seconds worth of someone talking or 60 seconds of music. And you get this from deconvolving. And the stimulus here we're using is not the acoustic representation of the stimulus, but it's an auditory nerve model representation of the stimulus that comes from Laurel Carney's lab. And what's fascinating and potentially extremely useful is this is still characterized by the same three robust peaks. There's an early one, there's a middle one, and the late one, which late one here meaning like 7 to 9 milliseconds, which correspond to the 3 peaks that we saw in response to clicks. So this doesn't mean we don't have to use clicks to get these ABRs, but it does mean we don't have to use clicks to get these ABRs. We can present continuous speech and use those for anything else we want, in cortex, below cortex, higher order cortex, and still get these ABRs for free, essentially. These are results from Russ Maddox's lab. What is the temporal response function? Well, first we'll talk about temporal response function as a mathematical abstraction, then we'll get to what it actually is as a measurement. Temporal response function as a response to a punctate stimulus is just the shape of the response of that stimulus. And if you present that stimulus again, then you'll get the same response a moment later. What makes it different than just a response is that it's linear in the sense that if we have 2 stimuli, punctate stimuli that are near each other in time, then the response we'd actually see is the linear superposition of the first one and the delayed version of the first one. So they linearly add. That's why I refer to this as a mathematical abstraction. Obviously, the brain is not going to be this linear. The other part of linearity is if that we have a smaller response, smaller stimulus, we'll get a smaller response. And if we have a negative version of the stimulus, we would have a negative response. This is all part of the linear abstraction of a TRF. And finally, if we have not punctate stimuli, but if we have a continuous stimulus, then we'll do the same thing, but we'll do this at each time step. And so we will just add what the stimulus is and then the response to that, and then what the next stimulus is at the next moment and the response to that. And we'll get a continuous response to a continuous stimulus, but they're related by this TRF. Okay, so why is this useful? This is useful because we can take a response to a known stimulus, we can take some representation of the stimulus, and then we can ask, if this is caused by a TRF, what is the TRF that this gives us? This can be estimated by a number of methods, but no matter how you do it, you will have a predicted response, which is the convolution of the TRF with the stimulus. That's the linear operation we're talking about, and the actual response. And we can do several things with this. We can measure how good this is. We can measure the goodness of fit between the predicted response and the actual response. We can also look at the TRF itself and see whether it has interesting properties, like, for instance, what we just saw, that the TRF to this auditory nerve representation of the stimulus looks surprisingly like the response to plain clicks. So even though the brain is not linear, it doesn't mean that these TRFs can't be extremely useful. And then again, these three waves, these three peaks, we can map those back to in this case, the subcortical areas in the brain, and talk about different activity that's going on in these different regions. This is a brand new technology, meaning it's only a few years old that we're doing this with high-speed EEG recordings. Ross and a colleague of his who's a former student, former postdoc, Melissa Polanenko, and I have just proposed this to ARO as a special session. We hope it will get accepted. One can look at other areas, for instance, the thalamus, the medial geniculate nucleus, or medial geniculate body, MGB, is part of the thalamus. That's an auditory area. And that contributes to, like the ABR, it contributes other peaks that have different latencies. This set of peaks that's a little later are called middle latency responses when they refer to the responses to clicks. Responses to the thalamus and to early cortex, because actually the MLR, the middle latency response, is believed to be due to a mixture of thalamus and cortex. With EEG, it's a little hard to distinguish which is which. It's how cortex responds to clicks is not so interesting, but this is how cortex responds to continuous speech, and that's potentially very interesting. And this is some data that was recorded from our lab. This is Carl LaRude, who's was over on the side, and he does have a poster here today with this data if you're interested. This is the data here is not just the response to clean speech, but it's also the response to speech with competing speakers. So we can look at what's happening in the early and the later responses for speech and masked speech as well. Just moving things up the auditory chain a little bit. We can also, our lab has also looked at fast cortical responses in the same TRF framework. So this is responses to speech, 60 seconds worth of in this case, it's clean speech. This is with MEG now. I moved to a different modality. MEG is very sensitive to cortical responses. It's not responsive. It's not sensitive to subcortical responses. And here we can see that if we band pass the envelope in the 70 to 100 hertz range, instead of before we were looking at very high frequencies, and typically TRF analyses, TRF framework analyses use say 1 to 10 hertz or 1 to 40 hertz. Now we can see very nice responses, 40 milliseconds, that's late for brainstem, it's early for cortex. So this is consistent with primary auditory cortex. You just have to go faster to see only this primary auditory cortex. There's nothing later. Also, these are all positive because these are neural currents that are being represented here, and the neural currents can point in any direction in 3D. So we're just looking at the magnitude of those currents. The multiple peaks? Yes, that's because of the band passing. Yeah, so that's just this filter is between the actual stimuli are bandpass between 70 and 200 hertz, but the brain is really bandpassing between 70 and 100 hertz. And so this is ringing at about 80-ish hertz as a result of that. Other people who look at this have a cleaner version of this. You can look at the envelope of that instead, and then it goes away. And it also then shows when the peak is much, much more nicely. This is a little bit more raw. Yeah.
Are you able to separate out primary, secondary, tertiary areas of this?
At these fast rates, at around 80 hertz, this is the only response we see. We don't see multiple areas. So because MEG, like EEG, is sensitive mostly to the dendritic currents going into cortical neurons, for MEG, it's mostly cortex, then we interpret these as the thalamic inputs. And thalamus does not have trouble going up to 80 hertz, at least if you're looking at many, many neurons. Cortical neurons in A1 don't have any trouble phase locking at 100 hertz. They just can't fire very often at 100 hertz.
So if you change the window, slide the window down to lower frequencies, would you see longer latency?
Absolutely. If we slide down to below 40 hertz, especially down below 10 hertz, we see multiple peaks with different polarities. Yeah.
Yeah.
Sorry?
You have to bypass 80 hertz, whatever.
Well, if you don't, then these are small compared to the typical TRF responses. So this is about the same latency as the P1-like response, and this is small compared to that.
Because I mean, from a conceptual point of view, it's strange to model the system, the linear system, but actually the system that you're modeling is the brain plus the token behind.
Well, so this is extra information. So if you don't bandpass it, then you effectively have the natural 1/f bandpass of the brain anyways. So if you have 1/f, something that's at 80 hertz is going to be very small compared to something at 10 hertz. And so it's just hard to see this otherwise. It doesn't mean that you couldn't see it without doing a Fourier transform or doing a high pass of that signal later. But this allows one to see it later. And hold that question until I show you the next slide. Because here's the response to a cocktail party version of the stimulus, where there's a male speaker and a female speaker. And in the top one, it's the male speaker being attended to. In the bottom one, it's the male speaker is being ignored. It's the female speaker that's being attended to. And you can see on the right that the attended, the amplitude of these TRFs is significantly greater, and in fact greater in the majority of cases than the ignored one. And because this is in primary auditory cortex, we're not mixing this up with other things going on. Certainly at longer latencies, one can see that attention is important, but it's very hard to see attention being important like this at 40 milliseconds. And this is the only way we've ever seen to do that. But now I'll take your other question. Oh, that's completely unlabeled, is it? It's the same amplitude as on the right, but it's the peak amplitude. Yeah. I don't have a mouse here. It goes off my screen, computer screen to the right here, but it's not showing up here. So But yeah, it is the peak. Yeah, and also you can see this dotted line, which is not the peak, but it's the same height in both. So you can see that in the attended case, the dark wiggles are much closer to the dotted line than they are at the bottom.
So, Jonathan, this seems like a pretty small attention effect. And I'm just curious, if you set it up a little differently, so that there was a particular carrier frequency that you had to attend to or not attend to, would you be able to optimize the attention effects here? I mean, it's great that you're looking at speech, but I'm just curious. But if another design might clarify, exactly what the mechanism might be by focusing on sort of the.
I think that your work has shown that attentional modulation in primary auditory cortex is small. It's significant, but it's small. And so in your work, there's a distribution that shifts significantly to the right, but you have to squint and you have to then see that there are stars next to that shift. To me, it's amazing that we can see this non-invasively at all. I'm sure it could be optimized in some way, but to me, seeing the effect at all is a big plus.
Shows there's probably not much at all A1 in humans. And so maybe this is not even A1, maybe this is a hint of maybe secondary or something, because in A1 is really weak, as you said.
Yeah, it could be it could be non-A1 areas. It has to be basically the same latency as A1. And there are other, I mean, first of all, there's 3 A1s in human auditory cortex, but there are also other areas that receive very short latency inputs, and it could be one of those as well.
Just getting back to the actual measurement, and not quickly about, how big or how small it is, I'm just curious, for this measurement that you're showing a 10 mil ignore band, are we just supposed to look at the highest point in the curve and compare the highest point?
Yeah, so this is the grand average being shown on the left, and on the right are the peaks of individual subjects.
Individual subjects, were you looking at just peak amplitude or
Yeah.
all of it?
Peak amplitude, not area under the curve. Yeah.
There seems to be a latency difference between attended and ignored. Is this signify something?
So there seems to be of what? Say it again.
The latency of the response? See?
Yeah, the latency is not significantly different, but it doesn't mean it's not different.
Does this?
This is not a large number of subjects. This is, I think, 30. If it's not significant, then I'm not going to read into it because I don't have any hypotheses about it. Back. Yeah, I mean, 40 milliseconds is certainly enough for there to be cortical thalamical loops going on. I mean, the earliest cortical responses are are certainly before 20 milliseconds, maybe 15, maybe even earlier. So there's this, and this is in an intact behaving human. So whatever feedback mechanisms there are, they all could have taken place here. But
in the TRF extended has more peaks close to 50 milliseconds, the TRF ignored. Does the TRF shape imply anything?
yeah, I don't think the TRF shape implies anything. There's a lot of variability in the shapes for individuals, and you can even see there's a lot of variability on the right in the amplitudes.
One quick question. The variability for these really big changes is like 3 or 4 individuals, because it's a striking change in 10 and more. Were you able to correlate that with performance at a behavioral level? Like how well they were able to attend to a given?
Not for this.
It was intriguing.
Well, to let you finish your talk.
Okay, thanks.
Continue to talk.
I should mention that this last one is Rashab Kumari's talk, is also here and also has a poster, but it is not on this.
Skipping one thing.
All right. All right, so now we've finished the introduction. And I'll try and speed up a little bit. Categorical perception. This is the idea that if you, here's an example of it. If you linearly change acoustically the word bad to dad, and you look at the proportion of subjects that report whether it's bad or dad, then as you linearly go to the right, it doesn't change at all. The first step, it changes a little bit. The next step, at the middle, it's in the middle, but after that, it's almost at ceiling. And so this is categorically, not quite universally, but pretty strongly universally either bad or dad. So linear becomes nonlinear in terms of perception. This data was taken from rhesus monkeys. This was not taken from humans. So this is an example of categorical perception of non-same species vocalization. So it's a pretty general phenomenon.
But you gave him a categorical choice, right?
Yes, yeah. But it didn't have to be so sigmoidal. It really did. Had to be something. Hold the thought. Let me show you some more data in a few slides. For the monkeys, this is Yale Cohen's work. I didn't do any of this.
Because the definition has both this shape plus the discrimination shape.
Yeah.
Which repeats them.
Yeah. So I don't know that. I don't know the answer to that. But perhaps it's in that 2011 paper. Okay, so categorical perception adds robustness to communications. That's a clear evolutionary advantage to why an animal might have categorical perception. This also means that in practice, because we have it, categorical perception is pretty robust. So for instance, here I have three quite different acoustic sounds. They're related, but you can see that they're not only different lengths of time, different durations, and they have some similar characteristics, but they don't look like they are simple versions of each other. And yet each of these is the same word, help, just spoken in different contexts by the same individual. This isn't even across subjects. And so the fact that if you were to listen to these and you would perceive them as the word help in every time aids in your side of the communication as the receiver side in this being robust. So going back to the bad dad in these rhesus monkeys, Yale Cohen was also able to record from several different areas, but specifically in the anterolateral belt, and showed that in the neurons recorded there, those neurons also showed the same categorical processing, that there were a group of the bottom, I can't tell how many colors there are, but the bottom 3 or 4 and the top 3 or 4 are processed separately, quite separately, distinctly in terms of latency. So you can even see where there is a neural correlation of this behavioral measure going on. All right, let's go to humans. It would be really nice to record responses to phonemes, like the phonemes in help, as opposed to the sounds that cause the phonemes. The problem is that they are correlated, and so it is tricky to separate them out. You certainly can't do with evoked potentials because of the correlations baked in. But if you do multivariable regression in a time domain, you can start to piece these apart. And so that's what we do. We use multivariable temporal response functions, or MTRFs. So we can look at TRFs due to multiple representations of the stimulus, for instance, acoustic features versus phonemic features. And then from those, we can come up with different TRFs that correspond to those different representations. This isn't a magical solution because the correlations are still strong, and even MTRFs have trouble when the predictors are too correlated. We can sidestep this problem a bit by looking at measures of phonemes that are less correlated with the acoustics. And there are several good ones of those. There are some that are based on linguistic statistical distributions, such as phoneme surprisal, phoneme cohort entropy. And I'll show you what some of those are. Phoneme surprisal is essentially how surprising or how unexpected a phoneme is in the context of the previous phonemes that have already been presented in a word. So if the word starts with the phonemes k, then if the next phoneme is the one that corresponds to the letter M, that's not very surprising at all, because 45% of words in a very large database of English words that start with K have an M sound next to it. Most of those 4 words, most of the instances are from Came. A few of them are from Cambridge. There are 13 words in which it's followed by the phoneme corresponding to the S sound. Most of them are case, but there's also caseworker and casein. You get less, more surprising with cake, and even more surprising with cane, etc. And so we can look at this because this depends on the statistics of phonemes in the English language and not what the sounds are, just to do a measure of phoneme processing in the brain. That's the formula on the bottom for those interested in the actual math. Cohort entropy also comes from the statistics, but it is quite distinct from surprisal. It's how unpredictable the current state of the word would be. So for instance, if we look at the phonemes l, a, late, then because 95% of instances in this database, that's just the word. 5% it goes on, it's lakes. But if the brain were to just say, look, let's just assume it's lake and fix anything later, it would do a great job. As opposed to cake, where it becomes a little bit less focused, or bake, where it could be baker, bacon, baked, bake, then that's a very high entropy situation. And doing a good prediction would be much worse for the brain there. And so we can use that as another measure that's based on phonemes, but that's uncorrelated with the sounds. So we're going to do these multi-TRFs, the MTRFs, using those measures. And I'm going to show you some results that come from this, this study, where we used 4 different stimulus types. The first one is speech envelope modulated noise. This is a common straw man. It sounds like it's speech because it's made from speech, but it's completely unintelligible. Does everyone agree that's unintelligible? All right, this is the sound, this is the same sentence that it was based on.
Spring of 1806.
Highly intelligible. And then the other 2 examples we used, non-words or pseudo words, are words that sound like English, but they're not words.
And from Thurber in Jehovah's side, my confidence, getting vilified, lavatories, breathing.
So that's very useful for analyzing phoneme processing, but it can't be used for analyzing word processing. And then the last case we used is scrambled words, where we took text from the same narrative that we used for the clean speech, but we completely randomized their order, put in punctuation to give realistic pauses.
Even for good reach here, living fact, which was plants, fermentation consequence.
I like to think of this as what happens when we experience going into a seminar, into a field that's not our own. You understand all the words, just not in that order. So we look at the TRFs from different predictors, different representations. The one on the right is the acoustic envelope. This is the standard workhorse for doing TRFs in the field. We see that there are 2 peaks. Again, these are the absolute values, but I put in positive and negative signs to tell you what the approximate polarities were. So this is a P1 peak, P1-like peak and an N1-like peak. On the left, we have acoustic envelope onset responses, which is basically a fancier version of the half-wave rectified derivative of the envelope. You do a diff in MATLAB or Python, and then you half-wave rectify that and use that as a version. The brain really likes onsets. It cares much less about offsets. So we use both of these simultaneously. I guess I should have said, yeah. So there's two latencies there. The first one about 60 milliseconds, second one about 120 milliseconds. The phonemic responses, we put in phoneme onsets just to soak up anything that comes from the fact that our phoneme surprisal and our cohort entropy are all put at the same time. So we want to just put in a constant series of ones to soak up anything there. We do find a nice TRF for the phoneme onset. It's very simple. For phoneme surprisal, we also see two peaks. An early one happens to be positive in polarity. The second one negative. The same thing for cohort entropy. We see an early positive one. The second one is negative. These are the first ones are about 80 milliseconds. So these are about 20 milliseconds later than the P1. This is awfully early. This is these are responses that have been dissociated to the extent that we can from the acoustics. Plus, we're already regressing out the onsets of the sounds. So these are something that are quite linguistic. So it certainly looks like there's something going on very early where a phoneme has been detected and is being processed as such. And then there's a second opportunity at about 350 milliseconds to figure out something went wrong and to go back and do something about that. And these we call N400-like. N400 is not usually associated with things at this low level. It's more associated with words or semantics, but it is associated with predictive coding. Sam Noor-Eddin has a beautiful work on this. Sam is in the back somewhere. Sam just came to the University of Maryland 2 weeks ago as a new postdoc. And if you have questions about predictive coding at about 400 milliseconds latency, I would love for you to talk with him about that. We can move beyond phonemes, talk about things that convey meaning, which in phonemes don't convey meaning much in English, in language, words do, but animal vocalizations are often used to convey meaning. That's why many animals vocalize. So for instance, here's another study from Yale Cohen's lab. There's 3 sounds that are transmitted by monkeys to convey information about the quality of their food. If they like it, but they acknowledge it's food, they have a grunt. If they do like it, then they have 2 different sounds, which are, they sound quite different, but they seem to have the same symbolic meaning. They seem to convey the same information. And they were able to show that there's an area of the brain, this is the ventral prefrontal cortex, where the neural responses don't care about the acoustics, but they care about the information being conveyed. So I'm not going to say that monkeys have words, they don't have words, but they do convey symbolic information, and there's a part of their brain that can be seen that that correlates with that conveying of symbolic information. Words often convey meaning in human speech. So we can add a whole bunch of other word level features. We'll put in a word onset and surprisal and 2 different kinds of surprisal. Word onset means a lot more than phoneme onset, because word onset is, how am I doing on time now? Okay, thanks. Word onset is actually much trickier than you think it is. It's not just that there is a pause at the beginning of a word. There are often pauses at the beginning of a word, but there are often not pauses at the beginning of a word. And so here's a fantastic example of someone trying to process the catalog in a library in real time, which is a perfectly reasonable phrase, but the different ways that it can be processed are numerous. And so you can see that it might make sense for the word boundaries to be anticipated, processed in real time based on context. So processed in real time based on what's gone on before, but also it might make sense that you have to go back and figure out, the brain has to go back and figure out what they were later. And if you've studied the brain a long enough time, you'll probably agree that it probably does both, because the brain is a big place. Word surprisal, this is a non-contextual word surprisal. It's just really 1 over the frequency of a word. It's how often this word occurs in a large database. 1 over that is the surprisal, and then you take the log. And then we have a different version of word surprisal, which is based on context. So in this case, in this one, we model the contextual surprisal with GPT-2, which is an older open source large language model. If you have the words, if you happen to find, then you can ask, what is the likelihood of a whole bunch of different words that might follow afterwards? And the nice thing about this open source model is you can ask not just what's the most likely word, but you can ask what are all the words and what are the probabilities, so we can get a contextual probability from that. The word responses to the same experiment also show a strong positive peak at a relatively early time, 100 milliseconds. This is 20 milliseconds after the phoneme response, which is 20 milliseconds after the first acoustic response, but still pretty early. And again, this beautiful, and here it's about 450 milliseconds, and 400-like response with a negative polarity, implying to me, and I think there's there is something to support this, that there is an initial processing which can't be weighted by too much extra knowledge. It can be related by whatever knowledge there's going in. So it can be predictive coding based on what is currently available, but it's not based on going back and inserting anything because it's real time. Plus, there's additional processing at 400 milliseconds, which is really a lot of time in the brain. That's enough for all different kinds of other regions with vocabulary information, speaker information, intent information to come back and make changes. And just to place these two in relative to each other, the word surprisal without context, which is just 1 over the word frequency, and the word surprisal with context are very similar in shape. They're TRFs, but the word surprisal that has context available is much larger. It is much more robust, and that's good. This is interesting, especially because word surprisal in context is not the same thing as semantic information, but it's correlated with semantic information. So I would never say that knowing the word surprisal gives you the brain processing something proportional to word surprisal is proportional its semantics, because I think LLMs can also do this without, I mean, we can argue about whether LLMs understand semantics or not, but in the brain, they're probably very correlated. And if you can see this, then it's probably some aspect of processing the semantics. So we've got these different latencies on the cartoon on the right. We've got this hierarchy of acoustic to phonemic to word-based processing that is bottom up because it's early, it's short latency. And then we also have this extra top-down processing, which allows other parts of the brain to come in and perhaps do error correction. And then I'm going to do something really fast, but I think it's especially interesting because it's about a potential objective measure of speech intelligibility. It would be really nice if we could detect whether someone was listening to speech and we could tell whether it was intelligible to them or not without us having to ask them. Because asking them and getting answers, A, takes a long time, B, is potentially very boring to them, and C, they may not actually care to give you the right answer. So in this experimental design, we use vocoded speech and priming. We play degraded speech, which is largely unintelligible. And then we play the clean speech version of it, which is priming, because when you play the second version of the second instance of the first vocoded speech again, it sounds much clearer. It sounds partially intelligible. So I'll play an example. Unintelligible? Was that intelligible for anyone? Okay. This is the clean version. The text is down here.
Where the five small chambers arrayed in perfectly symmetrical stars, a pentagram.
Now listen to the identical first stimulus. Partially intelligible? Yeah. Not to everyone. There's a lot of things that can make this less intelligible. One of them is the speaker and room conditions. Another is some loss of hearing. Another is if English is not your first language. All of these things can contribute. I'm sure there's other reasons as well.
Play the vocoded speech multiple times.
Sorry?
Vocoded speech in between, just play the vocoded.
We had a control where we played vocoded speech again, and then the clean speech.
So the vocoded speech, the middle.
Yeah, we flipped the second and third.
Measuring out here. So you're trying to predict whether it's intelligible to the person.
Well, so we asked them. This is not the control case. This is just the same case we showed. And so how clear was the speech? The first listening, the second listening, it got clearer. There also is some learning effect here over the course of the entire session. So we wanted to make sure that that wasn't a giant problem, and that explains some of the overlap here. The story, great, thanks. The story that they were listening to is a story about apples, the Johnny Appleseed, and it turns out most of the subjects could actually understand the word apple, even in the control condition, hearing multiple examples of that word. So what we're interested in is the neural representations of this. And so this is the word onset. We looked at word onset under the hypothesis that part of the perception of it being more intelligible is we can understand what the different words are. And if we can understand what the different words are, we can also understand where the words began and ended. Whereas the first time we heard it, it was just garbled speech. It might as well have been one band instead of three bands. There's no evidence of where the words begin and end. So here's the word onset TRF from a single subject. There's a strong early response that's positive. There's a strong late response that's negative. Here we've projected this onto a plane, so we do have the positive and negative aspects back. And when we look at the early responses, the peaks in the left hemisphere, not on the right. Left is more important for language processing. There is a statistical difference that the early peak becomes larger post-exposure than pre. But what's more interesting is in the late component of the response, it's almost twice the size. It's not just more significant, 3 stars versus 1, it's a stronger effect size. In fact, if you look at the beige piece next to it, which is the response to clean speech, the late response to post-exposure is almost as big as it is to the clean speech. So we're seeing that there is this neural evidence of word processing as this speech became more intelligible. When we looked at the acoustic responses, there were no change. When we looked at word surprisal instead of words offset, there were also changes. So this is my last slide. You don't have to worry, Malcolm. We've been investigating neural speech and language processing in humans, but there's also broader impacts. Of course, there's clinical applications to this. We can also ask questions about the evolution of language. The categorical perception and categorical neural processing of vocalizations and speech, when they're the same or different, we've been able to see that for speech in terms of phonemes and words, It is dissociable from the acoustics. It does provide new insight versus versus linguistics. And these are not available unless the speech is intelligible. And a lot of what I talked today about was inspired by a workshop that was held a few years ago, for which Jonathan Fritz was one of the organizers, on what animal communications can tell us about human language. Thank you.
So I didn't realize this when we set the schedule, but you and Laura are coming at this problem from opposite ends of the spectrum. You from the neural side, I think Laura more from the linguistic side. She had one question, so.
Where spatially these neurons are, is that, and with the different TRFs to show, is it all the same spatial location or spatial location?
The spatial location for these results doesn't change much from stimulus to stimulus. They're all from the temporal lobe. They're all more from the superior temporal lobe than other parts, but they're quite broad because it's MEG. And there's also a continuum of left versus right. For the acoustic measures, they're very strongly bilateral, a little right hemisphere advantage. For the language-based measures, they're more and more left lateralized.
That increase over time? You see like more left lateralization as you move forward in time?
In terms of latency,
Like, I'm thinking of that, maybe that 400 millisecond potential is in some sense maybe more left lateralized, but that's a big higher level, but this kind of question.
we haven't looked at that. It's a little harder to look at the location of the N400 because it's so broad. And maybe it's there, but I guess I should have just stuck with the question. We haven't looked at that.
So David Popo in chat asked a question I was also wondering about. And do you see?
Hi, David.
Do you see a difference in auditory versus visual prompting? So when I was listening to your example, I was purposely not looking at the prompt on the screen.
Mm-hmm.
And I had a very hard time understanding it.
You're one of my troublemakers.
I know I am. But David asked the question. I mean,
yeah.
Do you see a difference between verbal auditory prompting and visual prompting?
Yes. So visual prompting, I cheated. I gave you a visual prompt, and that does amplify. But I also only gave you one example. I didn't give you any practice. I didn't give you half an hour of this. When we're doing the experiment, we thought about having audio visual, only visual prompts, and would have liked to have all of them, but that would have tripled the experiment time. So we ended up with this. But certainly the visual really does add to the effect.